Okay, hi everybody. My name is Choon Kiat. You can call me CK. So I'm actually from Umami Bioworks, and it's my honor to be here today to present some of the work that I did on high-throughput clone screening system and the subsequent suspension adaptation of the cells. So just for you guys who is your first time hearing Umami Bioworks, so we're actually developing a technology platform enabled in order to enable traditional food companies to produce cultivated seafood. And so because we are developing a technology platform, so we would really want to validate our technology on multiple fish species. So the species that we are focusing are like, for example, Japanese eel, red snappers, and also tuna. So they are basically from the ETP list from IUCN. So basically endangered, threatened, and protected species. And that also gives us another reason because these are in high demand, and therefore it's easier for us to reach price parity due to their high price. And so our process will actually allow the traditional food companies to have a localized production where it is being consumed, so to reduce the carbon footprint, be able to save endangered species from extinction, but most importantly, to actually provide a healthy alternative, being antibiotic and also pollution free. And last but not least, to offer like a stable supply of seafood for the future generations. So as I mentioned, because at Umami Bioworks, we are developing a technology platform. So we actually work on many part of the processes, ranging from cell line development to inputs, meaning the media, to bioprocess, and even to food application, just to let our future customer to actually have a feel of what they can do with our cells. Okay, so back to the main topic. So I believe that most of you that are in the audience here will agree this. It is very time consuming and laborious to actually develop a very good cell line. And there's actually a lot of genetic variation and therefore translate to phenotypic variation even within the same individuals. And more so if the cells actually divide because of the accumulation of a natural mutation or spontaneous mutation. And if you follow this logic, then you will agree that one of the way or the simplest way to actually get the best cell clone is simply just to increase the throughput of a cloning selection system. So I'm gonna first give an overview of how the workflow will look like, and then I'll share some of the data that we collected along the way. So we started with getting a piece of tissue from Japanese eel, and then culture them as per normal in like the cell culture dish. And once the cells are stabilized, we will then perform high throughput single cell cloning system. So the machine that we use actually allows a gentle dispensing of cells of less than 1 cells per well in either 96 or 384 well, and in a very fast manner. So you can typically screen for like thousands of clones in one single experiment. And so there are two things that we are trying to select here. The first thing is actually the doubling time, because we want a cell line that is proliferating as fast as possible. And the second characteristic is actually to select cells that can differentiate efficiently into their respective cell type, like be it muscle or fats. So for the first characteristic, which is to get cells that proliferate very fast, that would be very easy because you simply just let the cells to compete with one another, and then you just select maybe the top 20% of the cells that reach confluency first. And for the second characteristic is slightly more tricky because I'm quite sure that you wouldn't want to develop or assess differentiation capacity for like thousands of clones. So therefore, the method that we use is actually to split the cells into two different groups. The first one, we will be using them for maintenance purposes here. And then the second group of cells, we will be using them for differentiation assay. So a typical differentiation assay will take somewhere between 3 to 4 days because you don't need to actually wait for the cells to be fully mature before you tell how well they differentiate. So once the cells are like differentiation assay is done, so what we do is actually we will then do a high throughput imaging protocol to actually screen for the differentiation markers. So, and once you get, like maybe out of the 200 clones that you get that grow fast, you might get like maybe top 10% that will differentiate well. And that is when you will then really take those cells and make them into individual cell line before you move on to the next step, which is to adapt them to suspension. And once they are adapted, then you will want to scale them to a spinner and eventually bioreactor. So next, I'm going to show some of the data that we collected. So these are like the muscle-derived stem cells that we got from Japanese eel. So the first thing before you do single cell cloning is actually to ensure that your cell type the cell type that you're looking for is actually within the population. So in this case, we did a differentiation assay to ensure that there are muscle stem cells as well as fat stem cells within this population. And for this is how a typical differentiation assay will look like. So you will get some of the clones that can differentiate well into muscle, some that will differentiate well into fats. And what we discovered is that unlike mammalian system, usually the clone will actually differentiate into fats and muscle to a different degree. And yeah, so for this today's purposes, we are going to just talk about the fats cells here. So what we did is we take this population and we subject them to actually single cell adaptation. So the method that we use is actually a two-step process where we first subject the cells to a static culture with enriched media. And once the cells are starting to get used to this suspension culture, we will then move them to shake flask to further adapt them to shaking culture. And usually what you tend to get is actually three outcomes. Either the cells will die, they will not adapt to suspension, or that they will adapt but with like they'll form this kind of aggregates or clumps, which is not ideal. So what we really want is like a true single cell suspension culture, which we think will make the downstream bioprocess more easy. So that's what we did. We take the suspension adapted cell line and we actually culture them all the way from shake flask to spinner flask to bioreactor. But most importantly, because the cells grow well, but we have to make sure that they actually do differentiate efficiently towards the adipogenic lineage as well. So we have done, so this is just 5 days of the differentiation while in suspension, and the staining is BODP stain. So I hope you can see that most of the cells actually have the lipid droplet within them. And it is also important to ensure that the cells, the differentiated fat cells, they actually have similar fatty acids profile compared to the actual muscle tissue. So it's a bit hard to see, I think, but the top line is actually represent Japanese eel muscle. The middle line is the preadipocytes, while the bottom line is actually differentiated fat cells. So just a disclaimer that this is not a quantitative analysis, it's more for qualitative purposes. So the take-home message is that in terms of all the important fatty acids for fish, such as omega-3, like DHA as well as EPA, so they are actually present both in the Japanese eel muscle as well as preadipocytes and differentiated cells. So that's something that I think Mark Post pointed out this morning, that in terms of profile, they look similar, it's just that the absolute content is a lot lesser. So so for the future plan, so I'm not sure how many of you actually know this. We actually recently launched 3D printed fish. Is a world first in collaboration with stakeholder foods. But unfortunately, due to the recent event, they couldn't be here. But they are actually great partners, and with their 3D printing technology, we actually developed this thing that was actually being consumed by like even the top minister within Israel. So the plan is actually to do the same thing with these fat cells as well, to produce unagi fillet. So the conclusion of the talk, the take-home message is that I've shown you guys how we actually use high-throughput clone screening system to select for the best cells or clone possible, and how we eventually actually adapt the cells to single-cell suspension, and also eventually scale up the production to the bioreactor scale. And importantly, also we showed that the cells can differentiate well after being adapted to suspension. And the fatty acids profile is actually looking very similar to the actual fish fillet. So for the future work, we are trying to rely on the 3D printing technology to actually produce the unagi fillet. So stay tuned for the further announcement. So last but not least, I would like to acknowledge everybody from Umami Bioworks, especially Dr. Paramesh, so who has more than 20 years of experience in fish cell line development, and Kaushik for helping with the scaling up process, Desmond for helping with the fatty acids profiling, and Sushen and Kara for helping in every part of the experiment. And last but not least, just want to stress that we actually open for any form of collaboration. So please feel free to reach out to us via this email, or you can talk to me in the conference. Thank you.
Thank you for this call. Questions? Yes. Maybe take this one.
All right. Nice work. Thanks. I had a question about the single cell suspension. It's for some cells easier to do than others. I tried to look into it and it seemed quite hard. Did you have any influence to steering your suspension to single cell instead of dyeing or aggregate?
Yeah, it's a very, very difficult process. I think a lot of people are trying that. I think it's the holy grail also in this industry. So I wouldn't say that we have figured everything out. It's just that I think you need a bit of luck. But our strategy is actually to be as gentle as possible, so that you actually give enough selection pressure, but not to kill most of the cells. Yeah, so therefore we do a two-step process where we put them in non-shaking culture plate, followed by a shaking culture once they are slightly more adapted. Yeah.
Thank you.
No problem.
So there were one more question.
Hi, thanks for a really interesting talk and great progress with the eel. How did you find looking for markers to characterize your cell culture with? Did you find it a challenge, or are there easily kind of well-known homologs to, for example, mammalian cell markers?
Yeah, so that's a very good question. So, for people who are working with less well-studied species, I think that's one of the biggest questions. So to be honest, we are very fortunate that at least the antibody for myosin heavy chain can be used because the gene is very big, so the chances of binding the right region is very high. But other than that, I think in terms of all the stem cell markers such as PAC7 and stuff, is very, very difficult. We tried almost 80% of the antibody on the market, and yeah, it's really difficult. I think one of the approaches is if you have enough resources, you can actually custom made antibody. Yeah, but
Thank you.
it's a big problem.
Thank you.
Rachel is asking online whether the cells have been adapted to a serum-free medium.
So in this case, no, but we are trying to actually adapt to that. So it's a two problem that's very, very hard, both serum-free and suspension adaptation. Yeah, so I think sequential will be nice.
Okay, thank you very much. And Inbar has also asked whether you the best clones that you had kept their proliferation rate throughout later passages as well.
Yeah, so I think for fish, we have slight advantage in the sense that the cells immortalize almost spontaneously. So it's not a big issue for us. So these cells has been, before we even do a suspension adaptation, there have been more than P100s in the culture. Yeah, and it's continue, I think, at least another P20 after suspension adaptation. Yeah, thank you.
All right, thank you.